Welcome back to my channel, and today in this video we are going to study about karyotype. So first of all, let's see about the chromosome morphology. So as we have already learned about how the chromosome looks like, so we know that it has a telomere and a centromere part. Centromere is the center part of the chromosome and telomere is the end of the chromosome. So this is one chromosome. This is duplicated chromosome, but still we call it as a one chromosome because we count chromosome on the basis of centromere. So in a human body, we have 2n chromosome. So 2n means this n stands for this one chromosome, which is actually two chromosomes joined by a centromere. This we already learned. So in chromosome, we have the central part, which is called centromere, and the ends are known as telomeres. Now, on the basis of the position of the centromere, we can classify the chromosomes as telocentric, acrocentric, submetacentric, and metacentric. So as the name suggests, meta, M for meta, M for middle. So when the centromere is present at the middle of the chromosome, we call it as a metacentric. What is submeta? Not exactly at the middle, but it is shifted from the middle position towards the end. Correct? But still it is in the middle, but not exactly on the middle. It is shifted slightly towards the end. Now, acrocentric means when the centromere is present towards the end, we call it as an acrocentric. Telo, T stands for telomere. Telomere means ends. So when the centromere is present at the ends, we call it as a telocentric. So metacentric when the centromere is exactly at the middle, telocentric when the centromere is at the ends. Now submetacentric when the centromere is not exactly at the middle of the chromosome but slightly shifted towards the end. Whereas acrocentric means when the centromere shifted towards the end. So these are the four types of chromosome on the basis of the position of centromere. Now on human chromosome, the short arm is designated by the letter P and the long arm by the letter Q. If you can see here in acrocentric, this arm is a short arm and this arm is a long arm, and it formed due to the positioning of the centromere. So the short arm is denoted by letter P and the long arm is denoted by letter Q on human chromosome. Now, what do we mean by karyotype? Karyotype means it is a complete set of chromosome of an organism. Now, as you can see here, the chromosomes in karyotypes, these are arranged or lined up in descending order of their size. These are all the chromosomes of metaphase. These are metaphase chromosomes that are arranged in the descending order of their size. And from which cells they are prepared? They are prepared from actively dividing cells such as white blood cells and bone marrow or any such meristematic cells. Now, for its preparation, first, the chromosomes are treated with a chemical called colchicine. This chemical actually it prevents the cell from entering the anaphase. That means it stops the cell division. It stops the process of cell division so that the cells get halt at the metaphase stage only. Once the cell division is stopped, these are chemically preserved and spread on the microscopic slide, stained and photographed. The photograph is then enlarged and the individual chromosomes are cut out and arranged in a karyotype. The preparation and staining technique have been developed to help distinguish among chromosomes of similar size and shape. This karyotype is of trisomy X. Why trisomy X? Here you can see this is X chromosome. How many X chromosome you can see here? 3. So that means this is a condition of trisomy X. So karyotype is a complete set of chromosome of an organism where the chromosomes are of metaphase chromosomes arranged in a descending order. To halt a dividing cell at a metaphase stage, the chemical used is colchicine, and then it is chemically preserved, spread, stained and photographed. Now coming to the banding techniques, we have 4 banding techniques: C banding, G or Q banding, R banding. For different regions of chromosome, that is constitutive heterochromatin, facultative heterochromatin and euchromatin. What is the difference between heterochromatin and euchromatin? Heterochromatin means these are very densely or tightly packed section of the chromosome, correct? Because of which this segment cannot get expressed. Why? Because these are so tightly packed that the enzymes, proteins or transcription factors, they cannot enter inside the region, and hence the expression of this particular segment cannot possible. So heterochromatin means very tightly or compactly packed segment of the chromosome for which the expression is not possible. Euchromatin means loosely packed segment of the chromosome which undergo transcription. That means the expression of euchromatin is possible. So this is the difference between heterochromatin and euchromatin. Coming to constitutive and facultative means, constitutive means this is that part of the chromosome which are always in a heterochromatin state. That is constitutive. For example, centromere. So centromere always remain in a heterochromatin state. Facultative means they are on and off heterochromatin and euchromatin state. Why? Because chromosome where it is present inside a nucleus. So nucleus is like a suitcase, very small space where a very large amount of DNA is packed. So if the DNA remains open always, they cannot get packed inside the nucleus. So it is like some regions of DNA remain in a heterochromatin state until its expression is needed. Once the cell needs its expression, this heterochromatin gets converted into euchromatin and it gets expressed. Once its function is done, again this euchromatin gets shifted to the heterochromatin state. So this is the facultative heterochromatin. That means on and off they are present in heterochromatin and euchromatin state. So to stain such different regions of chromosome, different banding techniques are available. That is C banding for, as you can see from the alphabet, C here and C here for constitutive. Easy to remember. G or Q for facultative heterochromatin staining, R banding for euchromatin. So now modes of stain binding. A stain can bind to DNA through intercalation, minor groove or major groove binding, or through external binding. Now these modes of binding, they depend on the type of interaction between the stain and the DNA, which can be covalent also or non-covalent also. Non-covalent interaction means hydrogen bonding, electrostatic interaction, pi-pi interaction. We already studied about these non-covalent interactions separately in biochemistry subject. So you can see about the details of such bondings. Correct? So whenever we add any stain to stain any part of the chromosome, the stain forms bond with the DNA molecule, which can be a covalent or a non-covalent bond. In case of non-covalent interactions, it can be of multiple non-covalent interactions working together to bind the stain with the DNA. So first such staining is Giemsa staining. Giemsa is a visible light dye that binds to DNA through intercalation. Now it is a mixture of two dyes. One is thiazine, another one is inosine, that precipitates over the DNA and hence stain the DNA. Among the two molecules, the thiazine dye is of small size. So it diffuses easily and intercalate with the base pair of the DNA. Once it intercalates with the DNA, then this large, slow diffusing inosine molecules that comes and bind with thiazine, and that's how they get precipitated over the DNA molecules, giving it a purple color. Now this precipitation, it is favored on a hydrophobic environment. Now from this Giemsa stain only we get G banding. So G banding, it is the most used banding method for cytogenetic analysis. For this, first of all, we need to pre-treat the chromosome. Pre-treat means basically we need to degrade the chromosome or digest the chromosome. So for that we use protease, or sometimes we can use hot saline citrate or any detergent or urea. After the pre-treatment, this stain is used to stain the chromosomal region of the DNA. So now in G band, we have something called positive G bands and negative G bands. So if we see the positive G bands, these are darkly stained region of the chromosome. Now when we are saying it is darkly stained, that means somehow it is favoring the precipitation of the Giemsa stain more. So when it is favoring the more precipitation of the Giemsa stain on its surface, that means the region is hydrophobic, because in the last slide only we learned that the dye needs hydrophobic environment to get intercalate with the DNA. So that means if it is giving a dark band, that means somehow the region is hydrophobic in that area and hence it can bind more stain with it. Now when the region is hydrophobic, that means somehow it is a region where the segment is very tightly packed. Because the segment is very tightly packed, hence internally it developed a hydrophobic area. So the tight region we call it as a heterochromatin. So that means positive G bands, because these are dark bands. Dark band means they are getting stained more. Getting stained more means somehow the environment around that is more hydrophobic. More hydrophobic means the segment should be very compactly packed to be more hydrophobic in nature, and the compactly packed regions are called heterochromatin. So that means positive G bands, they stain the late replicating heterochromatin segment, and heterochromatin regions are AT rich regions. On the contrary, we have negative G bands, that means lightly stained bands. Light bands means the region is less hydrophobic. Less hydrophobic means the regions are not compactly packed. That means these are early replicating euchromatin. And euchromatin regions are GC rich. In DNA, as we all know, DNA consists of A, T, G and C, these 4 bases. But all the bases are not present in all the segment in equal frequencies. The segments which are getting expressed or the coding region of the DNA are always GC rich, and the heterochromatin or the non-coding regions of the DNA are AT rich. So the negative bands are from euchromatin that are GC rich, positive bands are from heterochromatin that are AT rich. So the next banding is R banding. These are also obtained from Giemsa strain, and they also reveal GC rich euchromatin segment. So because it is giving the GC rich euchromatin segment, so which corresponds the negative G band, because negative G bands also gives GC rich euchromatin segment. Now here for the pre-treatment of the chromosome, what we do is, it is the process in which the chromosomes are incubated in an ionic solution at a high temperature around 87 degrees centigrade. After that, it is stained with Giemsa. The incubation process causes the denaturation of the AT region of the chromosome, because AT region has low melting point, approximately 65 degrees centigrade, as compared to the GC rich. So because for this banding, the chromosome is pre-treated in an ionic solution at high temperature. So because of high temperature, the AT region become denaturated, and hence what is remain is only the GC rich euchromatin. So R banding is for GC rich euchromatin. Next is the C banding. C banding, as we already saw in the slide, C for centromere, C for centromere banding. So C reveals the AT rich centromere, because centromere is also a heterochromatin state of the chromosome. So it is also AT rich. And what kind of heterochromatin it is? Constitutive heterochromatin. So what is the pre-treatment we give to the chromosome before Giemsa staining? That is involves acid strain, hot saline incubation and alkali treatment. What it do is, it depurinates the DNA and break the DNA backbone, which then cause the extraction of the DNA from certain region of the chromosome. As you can see in the figure, only the centromeres are stained or getting highlighted in this karyotype. So this is for the staining of only the centromere. Next, apart from Giemsa staining, what kind of stain the Giemsa is? It is a visible dye. Now we can also stain the chromosome using some fluorochromes. These are organic compounds that are capable of undergoing fluorescence. So in chromosome staining, fluorochromes are used when the chromosome are to be studied under fluorescent microscope. So although fluorochromes are less stable than visible light dyes, fluorochromes do not require any pre-treatment like Giemsa. So it has an advantage also, and it has one disadvantage also. The advantage is fluorochromes do not require any pre-treatment like Giemsa staining, and the disadvantage is these are less stable as compared to the Giemsa stain. So these are some of the fluorochrome that are used for staining of chromosomes, and different binding modes are given. For example, quinacrine is a fluorochrome which intercalates with the DNA, and they differentially fluorescence the DNA segment. So it is selective for AT. So that means they will stain only the AT. Stain means they will fluoresce. The AT part will fluoresce. So these are fluorochromes. So we can use Giemsa stain also. We can use fluorochromes also. If we have fluorescent microscope, then we can use the fluorochromes. Otherwise, we can go with the Giemsa stain. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.